talk about basically lineage diversity and how you decide whether a lineage is, you know, a substantive lineage, not subject to horizontal gene flow, and in that sense, it's on an independent evolutionary trajectory. And I'm not going to talk about species. I know Craig threw that hoary chestnut at me. I'm going to talk about lineages and diagnosable lineages, and leave the species question as to whether those lineages are species or not to another day, and basically just talk about diagnosable lineages, which could be geographic isolates or they could be reproductive isolates. So basically, the exciting thing for me is that PopGen and phylogenetics have come together with SNPs. So you've got these incredibly cheap genome-wide markers that are available to you that are immensely useful in PopGen to answer all sorts of questions there, but they also bring the phylogenetics discipline together with the PopGen discipline to answer questions about, you know, lineage diversity, what lineages are on independent evolutionary trajectories, and then also on species delimitation and also in phylogeography. So if you think about phylogenetics, you basically you're recovering a bifurcating tree to represent the pattern of ancestry and descent. So you're enforcing a bifurcating process on what you're seeing in the present, and so you're using the data from the present to recover what you believe is the evolutionary history of the species in the form of a bifurcating tree. And of course, that's not always the case, particularly not when you start to get close to the present. And so people tend to work with network analysis and throw that in under the heading phylogenetics. And so looking at, as Sally talked about and Craig talked about, you know, horizontal transfer as you come down the bifurcating tree needs to be incorporated into the analyses. And so there's a field of network analysis as well, which doesn't require the trees to be bifurcating. And you have spatially comprehensive sampling, as Craig mentioned, but there's typically in phylogenetics fairly low replication at each site. So certainly with mitochondrial DNA, you might collect one or two samples from each site and generate a mitochondrial fragment and use that in your phylogeny. But really that's typically, from a PopGen point of view, that's really low level intensity of sampling at each site. Phylogenetics applies from shallow to very deep divergences, and it's historically been restricted to empty DNA, but of course in the last decade that's been changing, and with the genomic revolution, you know, where you can generate whole genomes for your taxa, it's really rapidly changing. So that's just a qualification. In PopGen, you tend to look at contemporary and recent processes. So you're looking at the sort of exchange gene flow and lack of gene flow in a contemporary context across the landscape, and recent processes that may have occurred, you know, in the last 1,000 or so years, and you're looking to see what impact that might have in terms of explaining the contemporary processes. So you're not looking at South America and Australia in terms of a phylogeny in PopGen, but you would be in phylogenetics. You're also looking at population differentiation. So you're looking at how two populations differentiate in terms of their allelic profiles. And to do that, you need to get a handle on what those allelic spectra are for each of those populations. So it's sort of a different sampling regime that you're looking at. You need to sample a number of individuals. You know, probably 30 is probably a good target to get a handle on the allelic spectrum for each population. Look at population differentiation. You're looking at gene flow and admixture. You're looking at population structure. You know, are there cryptic lineages there that you weren't aware of that, within species that might be of interest in terms of defining management units for management or evolutionary significant units in terms of below species structure. You're looking at relationships among individuals, including kinship, and you're looking at population assignment. If you've got an animal in your hand, can you work out where it came from? That sort of population assignment. Okay, so they're quite different. And as Good Lord, can you read that? And there's a tutorial that goes with this, which is on fixed difference analysis, which has been uploaded. So there's a tutorial, but you can't run through it now. But run through it out of hours if you're interested in this topic. So it's called a technical note on fixed difference analysis or something, and it's got, it's a worked example type thing. Okay, so I'm only interested in lineage diversity today, not in species delimitation or phylogeography. And in particular, I'm interested in convincing you that a good way to determine whether a lineage is a substantive lineage is whether it meets the criteria of diagnosability. So by diagnosability, I mean if you take an individual in your hand, there are traits that individual has that can assign it unambiguously to a lineage. So it's not a statistical thing. It's whether or not the lineage is actually diagnosable by one or more traits. So all individuals in a group of individuals possess heritable character state that's absent in all the individuals outside the group. That's what I mean by diagnosability. Okay, and of course in the taxonomic world of morphology, this is well established. So when you describe a new species, for example, there's a section in there, diagnosis. And in that diagnosis, you have to state, what character it is that enables you to pick up an individual and say it belongs to that species. So it's been well established in terms of the morphological world for a very long period of time, but not so much in the genetic world. Though of course, you can have diagnosability genetically through a chromosomal rearrangement that has come to fixation and results in reproductive incompatibility. And that can happen very, very rapidly and not be accompanied by any sort of genetic divergence in terms of SNPs. So you can have a species that, it can be diagnosed by chromosomal rearrangement, but, the SNPs won't show you anything because it happened so recently. So, but, people understand that concept of diagnosability at that level. But in terms of the SNPs, the concept I'm going to get across to you is that fixed allelic differences are the fodder of diagnosability in the world of SNPs. And so basically, as you know, alleles wax and wane in their frequency in a population. Two populations can diverge in terms of their allelic frequencies. But when an allele comes to fixation, it's an irreversible event. It's the only way it can be reversed is through migration or convergent re-establishment of that allelic state. And so from a popgen point of view, it's an irreversible state in that you can't easily go back from it. And so it's a substantial biological event when you get a fixed difference occurring in a population. And so a fixed difference occurs when, if you've got two putative taxa and you take one locus and it's 100% one allele in one taxa and 0% allele in the other taxa, you've got a fixed difference, right? And of course the taxa that's got a 0% can't recover from that unless a migrant comes in. So it's a good indication of diagnosability. So you can pick up an animal, you can score it for that locus and say immediately whether it's in putative taxon A or putative taxon B. So it's diagnosable. And that's why I like fixed differences. So why is diagnosability important? It's because it can't be sustained in the presence of gene flow or admixture. So if you've got any gene flow or admixture, you're going to lose your fixed differences. It can't be sustained in the presence of that. So when you do have fixed differences, you can infer from that. If it's in sympatry, you can infer straight away that you've got two distinct reproductively isolated lineages. If it's in parapatry, where there's opportunity for gene flow, but there are fixed differences there, you can say the gene flow is not happening. And so you've got two lineages on independent evolutionary trajectories. And of course, in allopatry, the same thing. If you've got fixed differences there, you can say, well, they're geographically isolated, they're on independent evolutionary trajectories. And so it's evidence of that conclusion. And of course, if you're a scientist that works on progressive advance in understanding on the basis of evidence, absence of fixed differences means that you cannot challenge the null hypothesis that two lineages are actually one. And so you accept that they're one until evidence comes to demonstrate that they are two. So they might be two, but in the absence of fixed differences, you have no evidence to draw that conclusion, so you stick to the status quo. So it's a sort of hypothesis testing type approach. Okay, so diagnosability implies an independent trajectory, but of course something can be on an independent trajectory without being diagnosable, but you just don't make that conclusion because of Occam's razor, right? So I think that's important. The other thing that's important about these things is that you all know about hypothesis testing. So if you do a t-test and you get a significant result, you can walk away with confidence in that result, saying, well, there's a 5% chance that I'm wrong, but I'm going to wear that risk, and I'm going to conclude I've got a real result here. If you get a non-significant result in the t-test, it's ambiguous because you don't know whether it's because there was no difference there or because your sample size wasn't big enough to pick up the difference that was there. So it's ambiguous, and it's the same with these things. So if you've got two populations and you take 3 animals from each of them, and there's no fixed differences, no amount of adding new animals is going to change that, right? So you can be absolutely certain if there's no fixed differences between two populations, no matter how little your sample size is, there's not going to be a fixed difference generated by increasing the sample size. But the other way doesn't work. So if you've got low sample sizes, you're going to get false positive fixed differences. And so when you do get fixed differences, you have to test and find out whether those fixed differences could have occurred just spuriously as false positives based on the sample sizes, or whether there's evidence that there's a real fixed difference there. So it's a little bit tricky though. It's sort of the reverse of the t-test. But anyway, so under the hood, basically the way you undertake this analysis is that you sample comprehensively, and if you don't believe that, take a look at the paper by Chambers and Hillis in 2020 about what happens when you sample over here and you sample over here and you don't sample in between. You're going to call them taxa. You're going to call them independent evolutionary lineages when in fact they're not, because you haven't sampled in between, and so you haven't shown that there is actually gene flow between them. So you have to sample comprehensively, and yeah, I mean really comprehensively. And then the other thing is you have to collect multiple individuals from each site. So you need to get a handle on the allelic frequency spectrum for each site. So I think a minimum of 10 is good enough. 2n equals 20. You might push that down to 5 and still be able to use it. I think Mark would agree. You could probably get away with 5. But yeah, you can use 2, but so the reason that you get the analysis catches you out is because it does this. If you use 2, the false positive rate is going to be phenomenally high, and and so when you go to if you use 2, when you go to generate your independent lineages, everything collapses because nothing's significant. So it does catch you if you use 2. But if you've got 2 here and 100 here, then the false positive rate's still very low. So yeah, this is a target. Your target is 10 individuals per site. And then the next thing you have to do with this analysis is remove sites with evidence of admixture, because one sneaky little F1 hybrid in there will amalgamate 2 populations with this analysis. So you have to remove any admixture, and you probably should do that for phylogenetics anyway. Like you don't want admixed individuals in there. Individuals are hybrids between populations in your phylogenetic analysis because it stuffs it up. So there's nothing fancy about that. And then you use GLPCOA and GLPCOA plot to plot your structure, and you can usually pick out individuals that look like they're hybrids or integress, and you can use hybrids to identify those and then take them out of take those sites out of your analysis. And I'll show you doing that. Then you generate a phylogeny, and both Craig and Sally have talked about that, and Sally will give a bit more later. And then you do a fixed difference analysis to identify your diagnosable groupings, and you use geo-fixed diff and geo-collapse to do that. And I'll show you that in a sec. And then you overlay your diagnosable groups on the phylogeny, and you overlay it on the spatial representation, the map where you got your samples from. And by overlaying it on the phylogeny, then you can have a defensible means for saying, well, this clade is a diagnosable clade, and therefore it's a candidate to be called a species. It's certainly a candidate to be called a lineage on an independent evolutionary trajectory. So you're using the diagnosability to overlay onto your phylogeny to make your call. So here's an example of a group of turtles that are quite diverse morphologically. And the question is, are they different species? But I'm not going there. And the idea is that you sample comprehensively across a range. So each of these sites have at least 10 individuals per site. And then that gives you the fodder to feed through this fixed difference analysis. And then you remove the sites with evidence of contemporary admixture. So the Bulu River, between the Cooper River drainage and the Warrego, has a lot of admixture in there. It's really clear. You run the new hybrids, you can get F1 hybrids, the back crosses, the F2 hybrids. And so, there's a lot of funky admixture going on in there. So depending on your species concept, you might say, well, if there's admixture going on at the boundaries, they're the same species, in which case you'd leave this in and it would amalgamate the Cooper and the Murray-Darling. Or you might say, I'm quite willing to accept some level of hybridisation and integration at the boundary between two species, in which case you take the Bulu River out of the analysis. So it depends on your philosophy what you do. And of course there's funky admixture up there in a couple of other sites, so they were taken out. I don't know whether it's funky or not, it seemed funky to me, because one of them was admixing with something we couldn't find, but we found it later. But and to do this, you use the basically you use the combination of PCA and new hybrids to work it out and take out the populations that are suspicious. Okay, so don't try and follow this at the moment. There's a tutorial that you can do in your own time. Then you can plot these sites in a PCA and start to see what structure's there. There's really nice structure. The Lake Eyre Basin's over there. The Murray-Darling Basin's down. Murray-Darling Basin's there. Lake Eyre Basin's up there. You've got Burdekin and North all grouping together, and then you've got this sort of Cline running down from the Fitzroy all the way down to the Macleay River and the coastal east coast of Australia. So you straight away you're getting some feel for what you might expect to come out of your analysis, which is, I think, always important. Explore your data, get a feel for it. Then if something goes crazy, you notice it. Okay, and then of course you generate your phylogeny with SVD quartets, which Sally's talked about. And this is the phylogeny for all those localities. You can see there's quite a bit of structure there. You've got your outgroup here, so ignore that for the moment, but there's certainly a lot of, there's clades in there that you might say, okay, these look like they're lineages on independent evolutionary trajectories. You might say, well, these are species too. So, how do you make that judgment is the question. And people are making that judgment all the time, but how are they making that judgment? And so I'm arguing, let's go back to the fundamental concept of diagnosability to make that judgment. So you do a fixed difference analysis. And basically you just run geo-fixed difference, geo-fixed diff on your genline object, and it will generate a matrix of fixed differences between all your populations, all your sampling sites, right? And then having done that, you say, okay, I want to collapse all the sites that have no fixed differences. I want to collapse them into one aggregation. So it goes through and looks at all the ones that have got no fixed differences, or if you're a bit if you're a bit worried about that, you might say, well, you know, I'm going to say 2 fixed differences. If they've got, you know, no more than 2 fixed differences, I'll amalgamate them. You've got that option. But I tend to do it with no fixed differences to start with. And here you can see straight away that it's amalgamated all the rivers that are north of the Burdekin. So the Burdekin, the Ross, the Johnson, the Russell and the Barron have been amalgamated into one entity because there's no fixed allelic differences between the populations of those rivers. And then it's amalgamated all the Cooper sites, and it's amalgamated all the Murray-Darling sites, and then it's amalgamated basically the Fitzroy, Mary and Burdekin and Kohler into one. And it's amalgamated the Brisbane River south into one. Okay, so the first pass has already done a lot of amalgamation of these sampling sites. And then you run it again, and it takes the aggregations and any remaining populations, does it again, and then it joins the Styx River with the Fitzroy River, and they're adjacent, so that makes sense. And then you run it again, and it says no further amalgamations at a fixed difference less than or equal to 0. So you run it iteratively like that. And the really important reason for doing that is because this analysis will recognise a ring species, for example, and call it one species. It'll recognise a cline where you've got fixed differences between the top of the cline and the bottom of the cline, possibly really substantial ones, but the intermediate populations show that there is contemporary gene flow running down that cline, but not fast enough to eliminate the fixed differences at the ends of the cline. So it amalgamates that into one taxon as well. This is something the other techniques that are available don't do very well. So it's basically, you have to make that decision that you can't, if you've got a ring around the Arctic Circle where you've got reproductive isolation at one location, but gene flow around there, are you going to call that one species or two? If you're going to call it one, this is the technique for you, right? Because it will amalgamate them. So that's an important aspect of this analysis. The other thing is it also recognises that elephant in the room, that if you've got a species that emerges through selection from a parental set of populations, what will happen in your tree is those parental populations they become paraphyletic. You've got a species emerged from within. And the phylogenetic tree doesn't handle that very well. It'll suggest you describe the two parental groupings as species or lineages, when in actual fact they're not. This analysis won't do that. So it'll identify the one that emerges from the parental populations, but it keeps the parental populations as one lineage. So that's another sort of advantage of it. Okay, so here's our matrix. So basically you've got the matrix of fixed differences, and you can see that some of them are quite low. There's one there with 1. And there's one with 7, for example. So you might say, well, I've got 7 fixed differences, but given the sample size of those two localities, is 7 something that I might just expect by chance? So what the analysis does, if you ask it, is it'll calculate basically the number of false positives you would expect given the allelic profiles and given the sample size. It'll say how many false positives you expect with error bars around that, and then I'll say, okay, what's the probability of getting 7? If it's less than 0.05, you accept that as evidence that those populations have fixed differences. If it's 30, then you've still, even though you've got 7 fixed differences, you can't use it. And of course, the first 7 might be significant, and the second 7 might not be significant because the sample sizes are different, right? So you can't just look at the magnitude of it. And indeed, yeah, you can have quite substantial fixed differences, but if you've only got 2 animals from each site, then the false positive rate could exceed that. So you can't just look at the matrix. And so this has really held people up. People like my colleagues, Brad Schaefer, he didn't like fixed differences because you couldn't distinguish between the false positives rate and the number you were observing. And Mark and I used to have a rule of thumb, and no one accepted the rule of thumb, except the Australian Journal of Zoology. No, we had a rule of thumb. But so what I did was I did the maths and came up with a way of estimating the false positive rate based on some maths, which is in the back of the tech note that I showed you. So if someone wants to check that maths, I'd be really appreciative. I've tried to get mathematicians to look at it, but with little success. Yeah. I got a maths degree, so I think it's right, but someone needs to check it.
Thank you. I'd like to know what's the recommended level of filtering to get to the matrix of fixed differences.
Yeah, so you need to be careful because if you've got a false heterozygote, it will say it's got both alleles, and there won't be a fixed difference there. So if you're miscalling your heterozygotes, that's a bit of a problem. So you want to filter fairly heavily on read depth to make sure that your calls on your hets are accurate. But apart from that, I think it's okay with missing data. It will take that into account. It's not going to call joint missing. It's not going to call joint missing. It's not going to call missing and present. It handles that okay. So the normal filtering's fine. Yeah.
I've got a question. Would you not just filter for fairly high minor allele frequency to get rid of this problem? And one question, I know that's a bit picky, but you could think about the simulation that the situation that you have two populations, and one of them is the allele frequency is 0.9 something, yeah, and then it gets fixed in one generation, and that's the one you sample, and you have basically an additional effect that you have a fixed differences, which just was happened by drift.
Has to be fixed in both directions. So it has to be fixed in one state in this population and fixed in the other state in that one. Otherwise, it's just a private allele.
True. It's private.
So private alleles are different.
I'll take it away.
Yeah, okay, yeah. So it's quite difficult to achieve. I guess with the rare alleles, the way that we handle that is to reduce the stringency. So I think, Mark, you reduce the stringency on what's the fixed difference. So for me, it's 100 naught 100. I start from that. If I get a situation where it's becoming awkward and I analyse it, I can see why it's becoming awkward, I might move to 99 1, and you probably work at 95 5, right? 95 5. Yeah.
And I compare stringent 100 0 to 95 5, and that gives me a hint whether there's historical progression. The comparison for the value goes up enormously. You go to lower stringency. That tells you something realistic.
Yeah. Okay. So Mark uses 100 0 absolute fixed differences, and he compares it with 95 5, and that basically enables him to look for historical integration. That's I think what you said. You didn't have the microphone, so the online people, I'm just repeating it. Yeah.
Can I ask a question, Arthur? This is great, the fixed difference. I guess one question I have is around the time it takes to get to fixation is different depending on your effective population size. So things that are in a really large panmictic population might take longer to fix versus smaller populations. Has that been looked at all in terms of, because if you're looking at this across things that have different effective population sizes, does that matter or it doesn't?
It matters if they've both got low.
Yeah.
So they've got to both come to fixation. So if you've got two populations that are both low population size, they're going to come to fixation quicker. But the question is not how they come to fixation. The question is, once they've come to fixation, why hasn't it been obliterated if there's gene flow? It hasn't been obliterated, so there's no gene flow. That's the question you're addressing, not how they got to fixation or how the evolutionary processes that led to the generation of unique alleles that may have come to fixation. All that happens way before these considerations. All you're asking is there no gene flow in the presence of opportunity for gene flow? That's what you're asking, really. And so it doesn't really matter how they came to fixation. Though I can see, like the Hunter River, for example, was a bottleneck, and it's obviously lost a lot of alleles. And so, you know, the chances of the other population and it having fixed differences is higher. So, I mean, you just have to take that into account when you're interpreting it. Yeah. Okay, so you use GL fixed diff and you put test equals true to test it, and it does all the simulations. It takes quite a bit of time. And then you manually merge the populations that are not statistically significant with GL merge pop. So I did have it doing automatically, but it just, you know, you need to look at it. So it doesn't do it automatically anymore. So you have to merge them manually. Okay, so here's a tree. Which of these lineages and their descendant clades are on independent evolutionary trajectories? Well, here are the diagnosable entities. And so now you can say, okay, well, the clades that match these bars on the right are diagnosably different. You can take one animal from that area and you can say, based on the fixed difference profile, that it belongs to that clade. So it's doing what the morphologists want. Pick up an animal, what species is it? So pick up an animal, what lineage is it? And because the lineages are diagnosable and not just based on allele frequency profiles, which most of the trees are, this puts a nice overlay on it. And then, of course, you can then, once you've got your sort of diagnosable lineages, you can then map them on your PCA, and you can see that obviously it's making sense. so the North Queensland one's a diagnosable lineage. The ones from the Brisbane down to the Macleay River are diagnosable, but gee, that might be the one fixed difference thing. So you know you want to watch that one, and then the ones running down up to the Fitzroy River are a diagnosable lineage, and you've got the Hunter sitting out there like a sore thumb. And there's reasons for that. There's a portal that connects to the Murray-Darling Basin that floods in very wet periods, and the turtles have moved across from the Murray-Darling into the Hunter. So that's why it's pulled out from that sort of cline that you can see, a straight line cline. It's pulled out because it's had an influx of alleles from the Murray-Darling Basin. So, you know, it's interesting stuff, I guess, if you're into turtles. And diagnosable lineages, you can map them on your onto your map, and there you've got your nice diagnosable lineages mapped onto the map. You can hand that to the management agencies and say, look, these things are on independent evolutionary trajectories. Don't sell them in the pet shops from Adelaide and have them flooding here, because those independent trajectories are going to be disrupted by selling pets from Adelaide across Queensland. So, yeah, there's different reasons why they want to know about this. And of course, you've got that admixture swarm in the Boolloo, which is really interesting in itself. So the Lake Eyre Basin things are massive. The ones in the Murray-Darling are smaller. You know, size is not a good taxonomic character. You know, you're going to call them the same species, you're going to call them different species. So I've called them all basically subspecies within the species, like other people would call them all different species. So it depends on your perspective what you do. But everyone agrees on diagnosable lineages. There's no debate about whether it's a species or not, or a subspecies or not. You just sidestep all that. Talk about diagnosable lineages. All right, so basically we talked about lineage diversity. It's basically bringing phylogeny together with the concept of diagnosability, which is a sort of PopGen thing, and you get substantive diagnosable lineages at the end of the day. So it's a sort of a quantitative way of putting meaning on your phylogeny. Then you map it onto the landscape, and if you want to go through a worked example, there's this tech note, and it's got a worked example. The data, I've put the data up on the system here, so you can pull the data in and work through it and see how it works out. You can play around with some of the parameters. And yeah, I'll leave it there, I think. Thank you very much. Any questions? Well, is it? Yeah.
But having said that, I mean, the last bit of what I was going to talk about was actually just sort of going through stuff with the tutorials. So we've put all of that online available for people to work through, and you know, these analyses do take longer than this 2-hour slot anyway, but you guys can reach out if you've got questions. A lot of the stuff that's gone into the IQ tree, the SVD quartets, etc., has been pulled from, there's lots of great resources online, so we've sort of tried to allude to those as well, to if you want to go deeper and trial other data sets. So it's just small data sets that will hopefully run a bit quicker than normal to get you started. So I don't think we'll worry about the tail end.
Okay, well, I'll just point out that you've got two data sets. You've got the SNP data set and you've got the silico data set. Don't ignore the silico data set. So the agricultural people are right into it, but the ecological people aren't. And so basically you can do all these analyses with presence-absence data. And that's well established, and use parsimony. I think, is it Brian McGee? Matt McGee's right into this. I tried to get a hold of him, but couldn't. But he's right into this, and he's a strong advocate of using the silico data and using the parsimony techniques, conventional parsimony techniques for analysing the silico data. And so, you know, it'd be interesting to see whether silico data enables to go deeper into divergence than the SNPs, in the sense that the SNPs will start dropping out as you get further out, faster than the silico data. So this is something for people to explore and share on the data Google group if they have successes and things. So use that data Google group to, you know, communicate what you found and just get discussion going. Be really good. Okay. Well, I guess are we completely out of time? We've got time for questions for all of the speakers. Yes. Yeah, so questions. Just there.
Just a comment, not a question, related to going back to tree mix that Sally talked about. And I just want to flip the Amy's question, how deep you can go with tree mix, and make a comment about perhaps of how recent we can use it. Because lots of folks here have interspecific data sets, meta-populations, well-connected populations. And for us in the lab, tree mix has been extremely valuable to look at population relationships that are hardly separated over space. In a dramatic example, for the snapper, all around Australia, very well sampled. That's another point. Tree mix seems to work out for population level stuff when it's well sampled. We have an entire species with FST, global FST of 1%. And tree mix did a fantastic job in sorting out the stocks of snapper. And you get this drift contribution. You can even understand a little bit more of how each one of those stocks, which are management units, right, have been separated from others. And it's fantastic. I really like folks to perhaps considering playing with that. But it seems that high-level population sampling, like, you know, lots of individuals, lots of localities, helps to that. I don't know if anyone has any comment on how recent, you know, the in and outs of the software can let you go, but you know, our data sets have been really useful.
Yeah, so there's a GL2 tree mix if you want to use tree mix. I'm not sure whether you need to use it on a Unix box with multi-threading or whether you can just run it on your desktop. Just run it on your desktop, yeah. So GL2 tree mix will get you the input file for tree mix. So it saves all that fussing around trying to work out how to get my general object into a tree mix file. There you go, Cuddles.
Thank you. So just a comment on tree mix and then thinking about things in deeper time. I agree entirely with you, Luciano. It is a wonderful tool that's very quick. So it's modelling ancestral polymorphism and the effects on that of migration and drift, right? So it's not modelling accumulation of new mutations, it's just dealing with the stuff you had in the ancestral population. So it was originally applied to things like dog breeds, for example, and gave a lot of good insights. Now there's very recent separation and inbreeding. And then you mentioned fish population, human populations. So I think it's really good at that sort of population to phylogeography scale. I think once you start pushing up to where independent mutations are interesting, so it's based on genetic covariances among populations. So as soon as de novo mutation becomes a substantial input to that, you'll get less and less accurate results. So if you want to then go into network models, it's a computational nightmare generally. Because if you think of the number of parameters, with a nice tree we've got, even with species trees, we've got population size, divergence time, and mutation models. You add on to that reticulation events, with migration one way or the other way, the number of parameters goes up exponentially. It gets ridiculous. So there are a lot of people are working on this, these so-called network models, PhyloNet, PhyloNetworks, SNAC, you can sort of look them up. In our experience working with sort of exon capture scale data sets, it's been very, very difficult to get convergence in these very complex models. Once you go beyond 4 or 5 taxa, BPP is good if you give it a fixed tree. You can test type models, but I agree with you, tree mix is a wonderful tool.
You mentioned that when we have admixture, then we should remove those places. When we talk about admixture, do we mean 2 different species? No. Say, what's the difference of that with gene flow?
So basically, if you've got admixture, and you've got to make a decision as to whether you've got 2 putative taxa that are admixing, or if it's just admixture between populations that you would be quite happy to have amalgamated. But if you've got a hypothesis that there are 2 taxa there, and they're admixing at their boundary, and you're willing to aware the fact that 2 species even if different genera in the case of turtles, when they come together at their boundary, they admix. If you're willing to still call them distinct taxa or distinct evolutionary lineages, even though they are admixing at their boundary, then you would take those boundary individuals out, because the fixed difference analysis will just amalgamate them if there's any admixed individuals in there. So you have to make a judgment on that. So yeah, you don't want to. So if you got rid of all the individuals that had fixed, all the loci that have fixed differences between 2 populations, obviously that wouldn't do.
Is it possible to put the slides again? It's difficult. I had a question of that slide, that one. When you do the groups like Southeast Coast, I see like the tree is divided in one clade, and the following one it's linked to the MDB. So it's more like I don't really know how to interpret this, and
So you're talking about that little
Southeast
that little
Coast section. It has like 2 different clades, I would say.
The first 2 branching lineages, leading the Murray-Darling first.
Yeah, I mean that's a discordance. But you have to work through that. So maybe the tree's not right. So maybe have a closer look at that. I mean, yeah, they're 2 independent analyses, so there's no necessary reason why it's actually going to be concordant with all the clades in your tree. You're going to get discordance, and you have to deal with that in your study in whatever way you need to. So maybe I don't know. There could be reasons why the tree's wrong, or it could be reasons why the fixed difference analysis is wrong. So you have to work through that in your own study. They're two independent analyses. It'd be a major miracle if it always was concordant with the clades in the tree. Yeah.
I see. Thank you very much.
And so with the mitochondrial tree, you've got many more terminal taxa, and so you're going to get a lot of discordance like that, and so you're going to have to question each one of them.
Yeah, so thanks for the talk. So just with the admixture thing, the same thing that you were talking about, if they're not hybrid zones and if they're all in the same area, symmetry basically, and you suspect that there might be some kind of population structure, so would you, to remove the admixed, so you're trying to do a species delimitation with these two subpopulations that are in the same region. How would you go about removing the hybrids? Would you even consider removing the hybrids and then doing the fixed difference analysis, or would you what?
So
would you?
I would only remove the hybrids if you're in a hybrid zone boundary. Like if you've got hybrids and introgressions smattered through the two populations, well then they're not on independent evolutionary trajectories. So you would combine them. So don't start removing hybrids and introgressed individuals as smattered through the Murray-Darling Basin and smattered through the Cooper. Because if they exist, then clearly they're not on independent evolutionary trajectories because you've got gene flow between them, even though it might be episodic. So in the case of these, the complication is that the gene flow is episodic. So there are these portals between each of these drainages. So between the Warrego and the Paroo, for example, there's a portal that opens up every 5 or 10 years, Cutta Creek, right? And so, but there's some portals that don't open up for 10,000 years. But the signature of that gene flow exchange, that episodic gene flow exchange, will be present and will amalgamate populations that are isolated now. They're on independent evolutionary trajectories right now, but they haven't accumulated fixed differences because 10,000 years ago, the two rivers connected. So you have to interpret it all, in your knowledge of the landscape and your knowledge of the climate and knowledge of history and everything. So, but it gives you a foundation for working, not working with shadows. So you're actually working with entities that are diagnosably different, not entities that you've assigned importance to without as substantial foundation. Now, I know I'm in a minority in this. I've probably got a friend up the back. I know I'm in a minority on this, but I'm still pushing anyway. So, one little ugly fact. So science isn't a democracy, right? So, yeah.